I started my research path with a vision of a world where animal models are no longer needed in the development of new drugs and for drug testing. My goal is to help accelerate this transition. In our lab, in the Department of Biochemistry at Radboud EMC, we work with novel protein candidates that can be used for the treatment of many different diseases. I have focused mainly on working with proteins that can treat and hopefully at some point cure cancer. Cancer is commonly treated with chemical agents that are very effective at killing tumor cells, but they are not very selective, so they can also cause a lot of damage to other tissues, and we all are familiar with the side effects that chemotherapies have. Protein therapies can overcome this issue of side effects because they can be designed to recognize very specific proteins on the surface of tumor cells. So it's kind of if we had a Secret Service agent that is trained to recognize the good guys from the bad guys. In the body, therapeutic molecules follow complicated journeys to reach their destination. They normally are traveling through the blood vessels and at some point need to leave the bloodstream and start penetrating into the tissue. So if we think, for example, of our Secret Service agent from before, it's kind of like they're traveling via train and then they get off in Amsterdam Central Station. In our lab, we would like to be able to understand every step that these proteins take in this complex system so that we can improve them and make them better at recognizing only the tumor cells and not other healthy cells, so there will be less side effects. However, the traditional way to investigate a therapy is to do it in 2D in vitro models where the cells normally grow on flat plastic surfaces. And they are not grown in conditions that are similar to any human tissue. When a therapy shows some potential in these 2D in vitro models, then it is tested in an animal model. But very often, we don't see that the results that we saw in the 2D in vitro model are the same that we see in animal models. Sometimes, when the results in animals are promising, then they are tested further in human clinical trials. But we have seen also that these results from animal models are not very good at predicting the outcomes of the clinical trials. Since the current in vitro models are not complex enough to mimic the human biology, and we cannot really see inside animals to understand why the expected effects are not the same as in humans, then we need to create better in vitro models where we can have this complexity of the human biology and where we can investigate in real time how the therapeutic proteins behave in such a complex system. This way, we can improve them. For this reason, in our lab, we developed a tumor-on-a-chip system where we can replicate the complexity of a human tumor. In this tumor-on-a-chip, we grow tumor cells in combination with healthy normal cells, and we embed them in sort of a 3D gel. In the chips, we can also add the proteins labeled in red so that we can directly visualize them under the microscope. And we can directly follow how these proteins enter this 3D complex tissue and how they attach to the surface of the tumor cells. With these observations, we were able to then develop a mathematical model that could help us predict how these proteins would behave in a real tumor. We then tested the mathematical model to see if it was able to predict which dose of the proteins with the toxins would be better at killing the tumor cells in such a complex system. When we tried the predicted dose of the model experimentally in the chips, we saw that most of the tumor cells died and also that the normal cells were not affected by the therapy. So, going back to the analogy with the Secret Service agent, is kind of like they were correctly trained to kill the target without leaving any collateral damage. By showing how organ-on-chip technologies can be used to investigate the delivery of therapies in complex tissues, we are a step closer to reducing the use of animals at the early stages of drug development. Ultimately, we hope that this technology is so broadly applied that the use of animals for these purposes are no longer needed. I would like to be able to convince more researchers in industry and academia about the potential that organ-on-chip technologies has in the process of drug development, so that in the future, no animals will be required to test and develop new drugs.